Hello, and thanks for joining us. I am Tim Barnes. I'm the Operations and Commercial Director as part of the Environmental Science Division, and this is
Sebastian Mynott. I'm the General Manager and Founder at Applied Genomics.
And we're going to talk to you today about, we're going to give you an introduction to environmental DNA. We're going to talk about what it is, why it's important in the context of biodiversity, and also how to use eDNA and the data around it. So let's get started. There will be a Q&A session at the end, and as Katie says, if you just raise your hands, and we'll get around to them at the end of the presentation. I've just stolen my own thunder. We'll try and do the presentation within 25 minutes. It might be a bit more, but it might be a bit less, and then we've got plenty of time for Q&A at the end of it. So over to you, Sam.
Thanks. So as we know, biodiversity really underpins natural capital value. It's the fundamental unit of our ecosystems, which means it's a measure of our ecosystem resilience, its function, and its natural capital value. And these are factors which underpin our economic systems and our well-being. The thing is, most biodiversity is actually hidden from view. And so we need better tools in order to be able to accurately assess our natural capital. And I've been an ecologist for about 20 years, and the tool that I've found that works the best, which allows us to reduce reliance on indicators and measure biodiversity directly, is eDNA. It gives us an opportunity to use a high-precision tool that's cost-efficient for large-scale habitat monitoring. We can get deeper analysis of the risks and opportunities for our natural capital assets, and it enables early detection of the unseen risks of pollution effects and invasive species. So if you don't already know, eDNA is short for environmental DNA. It's the DNA that's left behind by organisms as they go about their messy work of just being organisms living in the environment. And what we can do is we can collect an environmental sample, and in that sample are the cells that are left behind by these living organisms. And then by separating out the organic material and then purifying the DNA inside that organic material, we get an understanding of the organisms that were living in that environment that we just sampled. The thing is, the whole process can look a bit like a black box. So the real question that we have is, how can we build trust in the eDNA data that we provide? And the key for us is to return to foundational scientific principles. We need to collect samples at environmentally relevant scales. We need to maintain accuracy with the appropriate quality controls. We can enhance detection with lots of replication. And we can use the genetic diversity data that we get to assess the population health. And of course, we want to ensure transparency in our monitoring, reporting, and verification. Now, the key things that we've learned along the way, and we've been doing this for about 10 years, are that sample size matters. So the greater the sampling effort that we put into looking for something, the more you look for something, the more you're going to find the things you're looking for. And that this should probably be the least controversial thing I say on that. We also know that metadata matters, and this is where our collaboration with Socotec is so important, is by measuring other aspects of the environment other than biodiversity, like the pollutants, let's say, that are in soils. We can directly contextualize the biodiversity responses to these environmental pressures. We know that quantifying the effort matters. So the way we take the sample, understanding that the effort that goes into the sample and not so much the volume that gets put through the sample. So the best example here is when we do a water sample, there are different ways to standardize how you take that sample. And you could say, well, we'll just take 1 liter every time and that'll be it. Or we can use a consistent sampling effort, as in that pump that we see there, which exerts a fixed amount of pressure and then draws out as much water as will pass through the filter based on the amount of pressure that's put onto the filter. And that is a more consistent way to calibrate the effort. We know that recording quality assurance matters. And at every stage of the way, the thing I like about eDNA is that we can quantify the uncertainties throughout every stage of the process. And then we can model them and we can report on them so that we can be very clear about how well we did detecting certain species in certain samples across a landscape. We know that repeatability matters. So if you're going to be doing multiple monitoring year on year, we need to be able to go back through the entire time series, through the entire data record, and be able to compare sequences that we have directly across these time periods. And that's only possible through repeatable methods for the data analysis that we do. We also know that transparency matters. So it's essential to provide access to all the data. So that is, identifying, where we found things and could identify sequences only to, let's say, the phylum level or the class level, and not necessarily just show a selected summary of the few species that came out.
Okay, so eDNA, as Seb just talked about, is the assessment and the detection of DNA from all types of species in whatever environment. And something to be very clear, although I might be known as the marine guy, eDNA collection isn't just for the marine environment. It's for soil and water environments, as you would do any type of chemistry testing. And this is what I wanted to speak about just now, is the sample collection can be, for eDNA, can be rolled in very simply, very cheaply, into your already existing chemistry sample taking. So if you're taking soil samples, water samples, or marine samples, marine sediment samples, if you're already going out to take chemistry samples, you can add another glass jar, which will be supplied by Socotec, and then you can send it back to us without doing anything differently. Soil samples, a small amount of soil, extra soil from an additional jar or a tin, if it is marine sediment. If these are treated appropriately and gotten back to the Socotec laboratory in time, we will do the filtering for you. We will go through the steps of doing it in the field subsequently in this presentation. But this can be a simple, cost-effective add-on to work you're already doing. And we'll talk more about the impact of the data in conjunction with chemistry, which makes already powerful eDNA data even more potent. So if you get the samples back to us, which Socotec will organize a courier, we'll be able to do it and make sure the quality and consistency of results is maintained, even if you're not sampling directly in the field. And the two different types of analysis that we'll talk about, I'll talk about just now, is the watery DNA and the soily DNA. So with the water, you're able to assess the microbial source tracking. So where is certain types of bacteria, what is the potential source of that, whether it's from certain species and even certain industries, and we'll get onto that more later. It's comprehensive, so you're able to monitor patterns within an environment, whether that's seasonal patterns or just any temporal pattern really within a river or marine environment. With the soils, yes, you're able to quantify things, but also with both of these analyses, actually, you're able to correlate them with chemistry data. So if you're already doing metals analysis, for example, or you're already doing organic pesticide analysis, or even flame retardant analysis, that tells you your industrial legacy. It can indicate you the current sources of chemical pollutants. When you combine that with the impact that it might be having on the biodiversity, you create a much more colourful picture as to what your data can tell you. And it's ideal for measuring biodiversity impacts along environmental pressure gradients and time. If you do this consistently for a relatively small cost, you can get a much bigger picture and be much more responsible with your environmental analysis.
Yeah, that's right. And just to run through a couple case studies about how that would play out in the aquatic water samples. So we've got assays where we can test for harmful bacteria in water samples. So in this example here, we've got a water sample which is tested positive for harmful levels of enterococci. But the key difference with the DNA-based analysis that we can do is that we can also detect the sources of that contamination, so that we can see the majority of that bacterial signal is coming from either humans or ruminants, and ruminants, importantly, not bovine, not cattle, but ruminants in the form of either sheep or deer or goats, that sort of thing. So all of that gives you a lot of information as to what are the potential sources and how to potentially mitigate those. As an example for the soils, the gradient that you can see along the x-axis there is pick your favorite thing that you're measuring, concentrations of metals or whatever it would be, and then we can assess what are the biodiversity responses in relation to changes in those concentrations in the soil sample. And what we're seeing here are certain populations of microbes and bacteria and invertebrates in the face of changing total organic carbon, for example, and we see certain populations which are decreasing in their genetic diversity, other populations that are increasing in their genetic diversity as that gradient sort of progresses. And then if you've got applications where you want to use one of our kits directly in the field, you can get in touch with Socotec. Socotec will organize for us to send you our kits directly, and then you can return that to Socotec. So you're still dealing with Socotec entirely the whole way through. And so it becomes very easy to be able to get one of our kits directly to your field teams. And it's very easy to use our kits out in the field. And so we've got two types of soil and sediment sampling kits. The eco-type is simply a larger volume kit for larger applications. The regen kit, smaller volumes for more high-resolution applications. We talked about the high-resolution application here, which is measuring along a transect or a pressure gradient in order to be able to see those proximal responses of the bacteria and soil microbes. With the eco-type, what you would be looking at is a more habitat scale. So you would have, say, a GIS mapping layer of all the different habitats that you're interested in across the landscape. And then you would take these larger soil samples within each of those habitats in order to be able to characterize what are the biodiversity which are most closely associated with each of those habitats. Bit of ground truthing, let's say, on your satellite remote sensor. Same idea with the waters. We've got the large volume in-depth eDNA sampler and the low volume easy DNA sampling kit. The low volume kit, super easy to use. Here's a couple of Muppets trying it out in a local pond, and very easy to be used by field teams to be able to collect a sample very quickly. And again, the type of outputs that we can get from that are microbial source tracking as well as other types of biodiversity monitoring. If you're going to get serious about monitoring biodiversity in the marine environment, though, what you really need is to collect a large volume of water over a significant period of time. All of our validation studies have shown that this provides the most robust result as far as being able to get a correct idea of what's going on for biodiversity in the marine environment. Originally, what we'd done is we designed it for early detection of non-native species in ports, for example, and we would set up an in-depth sampler upstream and downstream of a port. And we would use the tides to our advantage, where one sampler would be programmed to sample only on the incoming tides. And so the water would flow in from the estuary, flow across the port, and then get captured by that upstream sampler. And then that would go to sleep on the change of the tide. And we would use the tide in the other direction, where water would flow from the river, flow across the port, and then get captured by the downstream sampler. And so we end up with two data sets, one looking upstream, one looking downstream, and the intersect of those two data, the data that are shared in between those two those two samples gives us an indication of the species which are most closely related with the port infrastructure itself. The in-depth sampler really has shown its worth, though, when we're talking about open coastal waters, where we sample over a couple tidal cycles. And this gives us a very large-scale biodiversity profile over what is really a very complex environment. And it's only when you sample at this scale that you really get an accurate view of what are biodiversity profiles in open water bodies, tidally affected water bodies with lots of currents. And that sort of thing. The data that comes out of it is very interesting in the sense that we can get right down to species level in a lot of cases. And when we do, we're able to display, we're able to provide a quantification for the species that we've found, not the catch and count that you would typically get from your traditional monitoring methods, but a quantification of the genetic diversity that we find for each population that were detected. And what that gives us then is an indicator of the population health and resilience, which is arguably more informative than the conventional abundance estimates, simply counting which organisms are there. We also, as standard, provide extensive data quality reporting, and so we can give you all the reassurances that you need in order to be able to know that the species that we're detecting in the samples where we're detecting them, we quantify how well we've done with identifying those species in those samples. And then the return on investment. So larger samples, if you're taking the larger samples compared to the smaller samples, the return on investment tends to be you get about 7 times as much data per unit cost. And comparing eDNA to conventional methods, typically you've got a 5-to-1 cost to benefit ratio. And the added benefit is that in a lot of cases, we don't have to harm the organisms that we're measuring in the environment. It's a non-invasive method, non-intrusive.
Okay, so that's an awful lot of information to take in. So I'd just like to sort of summarize before we move on to sort of the Q&A session. eDNA is a proven technology that we can use to monitor biodiversity directly and more accurately than existing methods. You can scale your sampling up from small, localized environments to larger ones. That example that Seb gave about the incoming and outgoing tides, you think about it even in river systems, upstream and downstream of water treatment facilities, ports we've already talked about, but also maybe agricultural runoff, which I know I'm almost a Herefordshire boy in the River Wye, for example. It would be really useful data to be used in an environment similar to that. You can combine the sampling you currently do. It is way more cost effective than the catch and count. We'll talk more about that in a second. You have the more you do, like with any data set, and biodiversity data is no exception to this, is the fact that the more you do, the more reliable the data set is, especially over time. And we can provide you detailed quality reporting with credible analysis. And you can do all this in conjunction with Socratex sampling that many of you are already doing. And if you're not, give me a call. So we'll now go on to a Q&A. However, if you'll indulge me, I have a few questions about it, because I think some of the more sort of frequently asked questions. First one, Seb, sorry, these are all to you, mate. Apologies. So how long has eDNA been around and available?
Yeah, interesting. So the technology has been around for a few decades now, actually. We borrow the technology from microbial ecologists that have been doing the sequencing of microbes in soils and water for a very, very long time. So the technology is well proven. The difference is that now that sequencing technologies have come to the point where we can do it very quickly and very cheaply. We've adapted those methods to be able to look at larger organisms, eukaryotes in the environment. And Applied Genomics was one of the first to realize this. We started the company about 10, 11 years ago now. And so, yeah, we've been doing this pretty much longer than anyone.
Yeah, so although this is probably new information for most people listening, and it was new to me 12 months ago, I'll be honest with you, when we started working together, this is not new technology. This is proven. It's proven in the field. You'll see some of the case studies that Seb went through. You've been working with DEFRA, Natural England, and others. Another one, because I think it's useful for people who don't know, what did you describe the current catch and count standard procedure for biodiversity taken? Because I think when people understand that, they'll understand the potency of how much convenient this is.
So catch and count, I mean, it comes from fisheries monitoring, for example, where you would drag a net through the water for a certain distance, and you would catch a variety of fish species, and you would simply count them. And you would say, okay, well, if I drag the net over this distance over this period of time, and I've caught 100 fish, what's my catch per unit effort? And then you extrapolate that out across the water landscape. If I've got 100 fish in this net, that means I must have 100,000 in the wider environment or something along those lines. What we do is, rather than simply catching and counting the organisms, what we catch are the cells left behind by the organisms. And what we're detecting when we do our genetic analyses are the genes that are associated with each breeding lineage. So we can't count individuals in the same sort of way as you would if you were picking through a net. But what we are counting are the different breeding lineages that are in the population. And if we were going to extrapolate that out, as in how big is the population, if we know the species that we've detected, and we know what its average breeding rate is, we can simply multiply that out. If we have a certain breeding lineage, we have a number of breeding lineages, we multiply that out to what we think the average fecundity would be, and then we can make an estimate of the population size. There's no bigger leap of faith than in the catch and count.
And nothing dies.
And nothing dies when we do
that helps as well, when you measure biodiversity. So I know we've emphasized it, and if you'll indulge me, why is it so beneficial to do this in conjunction with other testing?
Yeah, so when we get a biodiversity data set, super interesting. It's great to see what species are in which samples are in which locations. All of that's really informative, but none of it makes as much sense as when we correlate that with the environmental metadata that we would get from a Socotec study, for example. And understanding, suddenly when you see the changes in populations, not just between places, but over time, and how those correlate with the changes in the chemistry, everything starts to fall into place and starts to make sense. Where are these pressures coming from? Where are the drivers? What do we need to do to correct those things to improve the biodiversity overall?
Fantastic. Those are my questions, Tom.
We have had a few questions come through. I don't know if you're able to see the chat.
Can I see the chat? No, I can't.
So first of all, we've got a couple of questions from Emily Burnett. Okay.
So I have a query about the types of your DNA assays Socotec can assess. Are there any biodiversity assays that Socotec cannot undertake, for instance, fish scrapes or You seem to know what that means.
So chondrichthyes as in sharks and rays, yes, we can detect those. They are the trickier ones to detect, though, so I will manage expectations. But yes, we do see sharks and rays coming through regularly on our fish eDNA analyses. I'm not sure what you mean by fish scrapes, as in, do you mean scrapes directly from a fish? Then what that would be is if you did a direct measure off of a fish, so getting a tissue sample or a fin clip or a scale of some kind, we could identify the genetic makeup of that fish, and then we could do population genetic studies directly from that. And that can only be done from individuals, not so much from the environmental metadata.
Okay,
There's a follow-up from Emily
yeah,
as
so
well.
there is a follow-up from Emily, and um, for regarding sampling kits, does the kit require specific training for field teams to use prior to going offshore, or is there an instruction manual for easy use out in the field?
So we do provide it. We've made all of our tools as simple as possible to use. They do require just a little bit of a run through essentially with your field teams. And yes, every kit comes with a quick start guide, which is a list of very simple instructions as to how to get it done. So even if you don't get a chance to speak to us, the quick start guide should answer all your questions.
And in fact, you've got some videos
We've actually got
for sampling
training videos
which
as well.
I've seen as well, which do very good stuff. So Michael Redford asks, understand that research has shown rapid deterioration of eDNA in samples not stored efficiently. How successful has it been to process 1 litre water samples in jars? Is 1 litre enough for all assays? See, turbid waters clog filters quicker, so we'll need to take 2 or 3 in some instances and ship these. Yeah, he's right.
Yeah, those are great points. And that's where I went back to the standardizing effort. Is if you standardize the amount of pressure that you exert on the filter, then whether you have a turbid sample or whether you have a very clear sample, the turbid sample you'll only get a few 100 mil through. The very clear sample you can get 2 or 3 liters through. Having the standardized effort means that you are consistently trying to pull as much water through as can be done for that amount of force. You're exactly right in that if you've got very, very clear waters, it's handy to have maybe 2 or 3 liters. But the key thing to understand is that eDNA is sticky. It tends to be in higher abundance in a sample where we have very turbid waters. So if you've got lots of suspended material in your water sample, you're also going to generally have a lot more eDNA concentration in that water sample. Very, very clear samples, you tend to have a lot less eDNA floating around.
Okay, so the next follow-up from Michael was, are the samples then being shipped to Applied Genomics? They are. So he's interested in how we found regards to eDNA quality following multiple freeze-thaw cycles.
Right. So what we're doing, the way we're doing it with Socotec is because we want to make it as simple as possible for people who are already taking samples in the method that they're used to with Socotec. You'll collect the sample like you normally do with Socotec. Those jars will come back to Socotec very quickly within 24 hours. Socotec will do the filtering within their processing facility. And then when they do that filtering, they will inject the filter with our DNA preservative. Once the DNA preservative is on the filter, then all DNA degradation stops at that point. And so we can be quite sure you don't need any cold chain after that. You don't even need to worry about time after that. The DNA preservative is very, very good at keeping DNA in its state until we get a chance to analyze it. If you are concerned about DNA degradation, then that's the opportunity to get in touch with Socotec. Ask them to ask us to send you a kit, and then you can collect the sample yourself in the field, and then add the DNA preservative in the field, and then send it back to us, and we'll do the analysis. But all of that can be done through Socotec.
And then one last one, apologies, from Michael. Can you provide bespoke assays if we submit, say, 10 samples and 5 are unable to be sequenced due to eDNA quality? How will this be costed?
Yes, I mean we do develop new assays all the time. We have people come to us with special requests. Can we detect a certain species? Can we look for certain groups of species? And it's a regular thing. We come up with new assays all the time. Happy to discuss that. How much does it cost? How long is a piece of string? It's the sort of thing that would need to be discussed, I think.
And then from Gillian, how are you determining the source of contamination, like humans versus ruminants?
Oh, right. So, well, there's a couple questions there, actually. So with the microbial source tracking, we're expressly looking for what are the different organisms that are in there by analyzing different types of gut bacteria, which are indicators for each of those species groups that we showed in that chart. If you're talking about contamination in a biodiversity sense, there are methods for taking both field blanks and laboratory blanks, so control samples along the way. And each of those controls then gets analyzed alongside all of the biodiversity samples. The information that we get out of the control samples is used to algorithmically decontaminate the samples. So we have a statistical method to remove any contaminants that may have been introduced throughout all of the processing steps.
There's a quick follow-up from Emily regarding the availability of sampling videos. They're not on the Socotec website at the moment, but we can send you a link to them. That's the problem.
There's also there's a Q&A box as well there.
Oh, is there?
So there'll be some questions in that.
Well, yeah, we will get to everything. Chris Ashleby mentioned quality assurance methods. So Chris Riley mentions in chemistry, we run proficiency testing schemes through Quasi-MEM, and also we're 17025 accredited. Is there an equivalent for eDNA at the moment?
So the only proficiency testing that there is for eDNA at the moment is in relation to great crested newt, and we participate in that every year, and we do very well every year. So so that's the only required standardization. There are discussions about other things that could be done in the industry in order to be able to standardize across samples. But what we do in order to be able to provide reassurance to our clients is we follow good laboratory procedures and provide all of these controls. And then we report on all of the controls, the outcome of all of these things, so that you can clearly see that we followed all the correct procedures, and you can see what the outcome is, how that affects the data that we deliver to you.
And on quality, because it's a rather embryonic's the wrong word, but new analysis in this field.
Yeah.
The quality assurance structures around it will grow with time, like they did with Quasi-MEM and all the others. Nicole asks, you mentioned longer sampling periods for more open marine environments. How long would you expect to need to take a sample in a mixed estuarine environment?
Yeah, we typically recommend at least one full tidal cycle, so that's 12.5 hours, or ideally 2 tidal cycles, 25 hours. So if you were in an open estuarine environment, you could drop the sampler in at 1 o'clock in the afternoon, come back at 2 o'clock the next afternoon, collect it, and then you would have your sample done.
Another question from, apologies for pronunciation, Sakthiviel. How is it applicable to all microbes, including viruses? If so, are we able to measure RNA viruses in the environment as well?
We have been doing a lot of testing. Actually, just this month, we've been doing a lot of testing on detecting certain RNA viruses in the environment, norovirus as an example. And so, yeah, watch this space, though there will be more to come.
Gillian asked a follow-up, what are the approximate turnarounds for eDNA analysis?
Right. So for the direct analyses where we're looking for just one organism, or in the example of that microbial source tracking, maybe we've got a few groups of organisms, but still these are very targeted sort of analyses. Those types of analyses can be turned around, like great crested newt, for example, that's another one. Those sorts of analyses can be turned around very quickly, usually within 10 business days, or if you're in a rush, within 2 business days. With the larger biodiversity monitoring type analyses, because of the sequencing and analysis that has to go on, our turnaround time is currently around 6 weeks. So we're hoping to bring that down.
We have a question from Joe James, rather long one, so I'm trying to summarize it appropriately.
Hi, Joe.
Yeah, Joe says, they're an advocate of eDNA is a complementary method for ecological monitoring, but thinks it's important to acknowledge the limitations of eDNA. Particularly the results we need to be used for informing management. eDNA does not necessarily prove the detection of species at the exact location the sample was collected. What are the limitations of eDNA?
Yeah, so I mean, Joe's exactly right. What we're doing, especially in a water sample, when we detect eDNA, we're not detecting the eDNA of an organism that has been at that location at that time. We're detecting the eDNA that is essentially floated in from somewhere. And that's why when we're talking about in the marine environment, sampling over a larger period of time, the reason we do that, the reason that we've shown that is actually the better way to do it is because eDNA will be floating around on these currents and tides. And if we're not sampling over that longer time period, as soon as you have a change in the tide, you get a very different biodiversity profile. And so, yeah, being able to sample over a longer period of time in a dynamic environment is what gives you a much better biodiversity.
And Joe also notes quite rightly, whilst the eDNA samples are relatively easy to collect, it is important because of the sensitivity of the method that careful decontamination processes for minimizing risk of contamination in the field should be implemented.
Exactly.
You're quite right.
And yeah, thanks, Joe. And that's also why we have the quality assurance reporting that we do, which takes into account the decontamination procedures and the risk of false detection. Essentially, is part of that calculation that goes into the quality report.
And that's true of chemistry samples as well. Certain containers are applicable for certain analyses. The way they're handled is really important. Michael says, in regards to reporting outputs, what can we expect our results to look like? Will we be able to provide fast QX files for all sequencing?
Yeah, so FastQ files can be made available, and we would make that available on our as through a file transfer protocol. You would download all the FastQ files if you want to check them yourself. Typically, what we send are a full data set, which is every sequence that we detected post processing and through across every sample in which it was detected. And the numerical values would relate to the number of sequencing reads for each of those sequences. We don't advise that you use the reads as a proxy for abundance because reads are biased, and so yeah, you're likely to get not entirely accurate results in that respect. And then we also quantify the taxonomic assignments for each of those sequences. And then we have a more simplified data set, which is our haplotype data set. These are all the organisms that were identified to genus level. Why to genus level? Typically, that's because that is the taxonomic level that most people would be used to using in other types of environmental surveys. And so that's the level at which it's most important for running through any other sort of ecological calculation.
Martin sent a question in, which I think is a really good question, actually. How far back in time will the eDNA report on? I.e., will the results include DNA analysis of organisms that were present 1 day ago, 1 month, or even longer? And will this skew the findings in that historical legacy cells might indicate the samples are still present when they are in fact no longer?
Yeah, no, that's a good point, and that's a strong point in soils and sediments, where it can be more of an issue than in waters. In waters, we know that eDNA has a half-life of about 22 hours. So the signal that we get out of water samples is relatively proximal. Where you have to be careful is in the sediments and the soils, because as you dig further down, especially in sediments, you can be seen to be going back in geological time, if you like. And so the way that we collect, or the way that we instruct you to collect samples in the environment is to essentially take a scraping off the very top of the sediment layer. And in that way, you're sampling the layer which has been most recently deposited by the organisms that are living in that environment.
I think we might have answered this one already, but Paul Rudman, could you elaborate on some of the current constraints of this?
Yeah, so there are a number of constraints, as has been pointed out before. One of the things that eDNA doesn't do is it doesn't count. We can't, in the traditional sense of, an ecologist would go out into the field kick over some rocks and count the number of bugs that they see under the rocks, and then use that as their catch and count sort of assessment of what are the population sizes of the organisms that are out there. We use, instead of counting individuals, we count the number of breeding lineages that we find in the sequences that we produce.
Michael Almond asks, can eDNA sampling identify mold species estimated concentration?
Yeah, so we can do molds and fungi very easily, just as easily as any other group of species.
And another one from Bradley Fisher Thomas. eDNA, we talked about soil and water testing. Is the technology available for air sampling, e.g. mold spores? There you go.
Yeah.
No, you've already done it.
Yeah. So we've got a few different methods for sampling those types of molds. So molds in air samples. So just get in touch and we can do that.
Fantastic. One more from Joe that's just popped up. Are you aware of any progress in the regulator approval for eDNA as a detection method beyond the great crested newt?
Joe, you must have been reading my calendar, because I've literally just come from a meeting with DEFRA, Natural England, JNCC, and others, exactly about questions around regulatory operationalization of eDNA. So getting it to the point where the regulators are approving it for project approvals and things like that. It's frustratingly still a work in progress, but it's also something that I know all of these agencies are working hard towards. So it's just a matter of basically getting to that consensus. And I think, it feels like we're getting very close. And so it's one of these sort of things.
Brilliant. And that's it from us. Thanks for all the questions. We anticipated a lot of questions, so that was good. I hope you found it really interesting. If you do have any questions that you just want to ping over, feel free to email us. In fact, I think there's the next slide. There you go. It doesn't have our contact numbers on.
That will be shared.
Because that will be shared afterwards, apparently. Thanks. But yeah, it's been a pleasure. I think that this is a really exciting new direction. I think we've done a lot of, obviously at Socotec we do a lot of soil and water testing in the marine environment, but this is another interesting thing that we can offer to anyone that wants it. Brilliant. Thanks very much.
Thank you. Yeah, it's been really good.
Thank you very much, guys. That was really interesting. Just to let everyone know that there will be a follow-up email, and a copy of the recording and the presentation will be available. If you do have any questions to direct to either Tim or Seb, we will also include some contact details in that email. So keep an eye out for that. And then in the meantime, if you do have anything else that you want to ask, I'll give it another minute or so. But I think from the looks of it, yeah, I think that's it for the questions.
Thanks very much, guys.
Thanks, everyone.
Yeah.
Thank you, everyone.
See you soon.